session that which is hands-on training on designing your custom guide RNA. So, as we have already seen that sometimes it becomes a challenging task for the researchers to design the correct guide RNA only, and because of which sometimes it can become a challenge to apply this technology in their projects. So I this is a little attempt from our side to let young researchers have a at least a beginner's understanding on how to design guide RNAs of their choice. So let's start. So before we start, I just want to take you all through some few 4-5 general tips or guidelines for designing guide RNAs. So first of all, so before you begin to design a guide RNA, the following must be defined. So first of all, you should know exactly the target region or the gene that you want to target, right? So it cannot be, you cannot be two ways about this, that you want to target a human cell or you want to target a mouse cell or you want to target a certain bacteria or a virus. I think it would be more wise if you spend some time on reading up about that target of interest and come down or narrow down to a certain region of the genome of that target organism, or at least a gene of interest. So that's very important. Then secondly, it is important to know that which version of Cas9 protein is going to be used for the experiments, including which PAM sequence has to be recognized. Now, this is very important because Cas9, as you can see, it can recognize 2 or 3 different PAM sequences. Similarly, the PAM sequence for CPF1 is different, or that for Cas12 or Cas13 is different. So knowing exactly which Cas protein you're going to use for the nuclease activity or for binding affinity, as well as knowing the PAM sequence, is very, very important. You can't you can't not know this beforehand. Then which promoter will be used for in vitro or in vivo expression of the guide RNA? Now, this is slightly tricky to understand. Now, the guide RNA that you have designed, it needs to be introduced into your cell. You cannot put it in the cell as it is, like you cannot inject a guide RNA directly, because if you do that, it will be attacked by the cellular endonucleases, exonucleases and everything, and your guide RNA will be destroyed in no time. So when you have to introduce this guide RNA into the host cell, it has to go in the form of a plasmid or in the form of a vector, with a vector. So it is very important to know that which promoter, genomic promoter, that you are going to use upstream of this guide RNA sequence, so that the promoter region has its terminator sequence. Now, if that terminator sequence becomes a part of your guide RNA sequence, your guide RNA will never get expressed inside the cell. So it is very important that you know exactly which promoter and thereby what is its terminator sequence, so that you can avoid having that sequence in your guide RNA. This is also these are basically some basic molecular biology concepts that you need to keep in mind. And finally, you have to also remember that what cloning strategy are you going to adopt, which might put additional constraints on guide RNA sequence, because sometimes you might have to put these restriction sites, or you might have to avoid certain restriction sites within your guide RNA sequence. Otherwise, if there is a restriction site in your guide RNA sequence somewhere in between, and you happen to use a restriction endonuclease thereby, it will get chopped off. It will never get expressed as it is. So this will impact the design of the specific oligonucleotides to be synthesized and either used as a template for RNA production or cloned into an expression vector. So you have to remember this. Now, all these 4 points that I have just mentioned in simplified language, it is also mentioned in the table which is given hereby, and it is adopted from this paper which I have hyperlinked here. So as I had told you about the version of Cas9, it is based on the approach of CRISPR-Cas that you are going to use. So these are some basic rules. Do we have some questions here? So
No, not at the moment. I can't see any
yeah,
questions.
because I'm on full screen mode. So if there is a question, Dr. Diksha, please let me know from the chat box.
Any
Yeah.
questions yet? And
Yeah.
again, iterate this. So, participants, if you have any doubts, please write your queries. So now we got a question. So why we are using cloning strategy?
Yeah. So just like I told you that in order to introduce your guide RNA as well as the Cas9 protein inside the host cell where you want to do the experiment. Let's say you want to do an experiment in mouse cells. So you have to transfect your plasmid which contains the Cas9 protein because it is not present in the Cas. I mean, it is not there in the mouse cells. You have to send it from outside, and that has to be done inside a vector for which you need to remember the cloning strategy. You need to know which exact cloning strategy you're going to use. Similarly, for the CRISPR sequence, which is not there in the host cell, you need to send your guide RNA into the host cell, for which you cannot just put your RNA or DNA there. It will be chopped off by the host cell nucleases. So you have to cover it up in the form of a plasmid or a vector, and thereby that vector has to be introduced into the cell, whereby by using the cellular transcription machinery it will be expressed inside the host cell. That's why cloning strategy is very important. Now,
Yeah.
so with that in mind, let's just straight away go to the features of the guide RNA sequence, which are important to be considered. So general guidelines for guide RNA design. When designing, so the guide RNA which we are going to design, usually it is a 20 nucleotides long sequence. It can vary from 17 and go up to 24 nucleotides, but ideal length is 20 nucleotides. So it's a short RNA sequence that has to be designed. Now, the first concern is the length that I've told you. Then the second one, although it is mentioned as number one here, because it is in the order of priority, the GC content. So the typical range of GC content is 40 to 80%, where a higher GC content stabilizes the RNA DNA duplex while destabilizing off-target hybridization. So as much as possible, we will always try to have a higher GC percentage so that the binding is more tight, or the hybridization between the RNA DNA complex is tight enough. Then, to avoid potential off-target effects of the, we have to make sure that the sequence is as specific as possible so as to avoid off-target effects. For this, it is very important that, even though there can be some mismatches in the sequence, we have to remember that the position of the mismatch matters a lot. Then number of mismatches. Imagine there was a sequence with only one mismatch, and then there was a sequence with, say, 5 mismatches. You would always prefer the one with single mismatch. Then sequence of guide RNA and the concentration which is going to get transfected. Although it is not a big concern in the beginning stage, but yeah, this is also a matter that needs to be considered when planning an experiment. So with this, we'll also try to understand what are on-target and off-target effects. So CRISPR-Cas9 can be very efficient. As much as 100% mutagenesis has been observed in some kind of applications. But this also comes with some kind of off-target, or that means side effects. Or in the common language, it is called as side effects, or effects or binding of the guide RNA at undesired sites, which you had not planned, but that just happened because of some kind of sequence similarity. Although it is not as much, I mean, if say 20 molecules are binding at the target site, you would not have an off-target effect where also 20 molecules are binding. Maybe only one will bind, but even that can have some kind of effect. And if that happens to be on an essential gene in the body or in the cell, that can kill the cell. So we have to keep in mind about the off-target effects also. Then off-target effects are thought to be most likely to happen at sites where sequence similarity is there to the on-target site, right? Now, so with this background, we'll try to have a look at some of the tools for guide RNA design. For a good hands-on learning experience, guys, please ensure that your internet connection is steady and you also have a paper and pen handy. Fine. So now we are going to go online and use online sources for designing your guide RNAs. So the first one shown here, I have hyperlinked the site here in the yellow bar on top, which is idtdna.com. So if you go and open that page, I've put the screenshot how it looks, and the steps are, shown in red colored circles. So when you open that page, you go to products and services, and when that page opens, you go to CRISPR genome editing page, and you click on that one, CRISPR genome editing.
Dr. Tani,
Once you do,
can
yep,
you share that link in the Q&A box?
okay, I'll do that.
Yeah, it will be easier for the participants to click there.
Sure, sure, sure. So
Yes,
I hope all of you got that.
yes. At least I got it. Yeah.
Yeah.
Well,
So again, so when you open that page, on the home page, you have this first tab, which is products and services. You scroll that down. I mean, you open the mouse over it and you will find the scroll down options. There you have to go and click on CRISPR genome editing. That is also clickable. Once you do that, the page opens and you need to really scroll down, and whereby you will find these kind of tabs. So you go to check your own design. And once that is done, this page will open up. So the red colored oval dotted where I've written number 5, that is a tab which you have to open. Design custom gRNA. Right? So once you have gone to that page, you have to select first the species of interest. Let's say it is Homo sapiens. That is also the default one. Then in the input format, you have to put the FASTA sequence. FASTA sequence of the target. Your target, either you can write the gene name or you can put the FASTA sequence, or you can also sometimes upload the file. So for the ease of this demonstration, I have got this particular sequence which I've highlighted in the top, which I have typed in the FASTA format. So what is FASTA format? So in the top line, you first write the name of the sequence. That is dependent on you. You can choose any name. Followed by, in the next line, you type the sequence or you paste the sequence. Fine. Once you have done that, you have to click design on the, once you have put the sequence, this bar becomes where it is written design on the right side, that becomes clickable, and you click that design, and then you just wait for a second to get this kind of output. So can I know a feedback from the audience if you are able to follow this far?
okay, so I am going to start a poll that if the audience has followed this. Okay, so
Please let me know if you've come this far or if you're facing any challenges before that this step.
at least I can see Tani 44% of them have completed all the steps. So there were few students who have not yet completed. So if
I'll wait for 2 minutes. Yeah, I'll wait for 2 minutes for all of us to, be on the same level.
yes, great. So if anybody is facing any issues, can we have a recap, Tani, once again?
Oh, sure, we can do that. So the first step is that you have to log on to the site called idtdna.com. The link has been pasted in the chat box. Please copy paste from there in your browser and you will go to the home page. So in the home page, there is a first tab, which is products and services. You mouse over it. So it opens a whole lot of options. In that, you scroll down and go to CRISPR genome editing. You click on that and then the whole page opens, whereby you need to really scroll down and come to the bottom of the page where you will find these kind of boxes. So the blue box where it is written check your own design, you need to click there. Once that has been done, it will take you to this kind of a page, input page. And in that, there is a middle tab, which is design custom guide RNA. You need to click there. Then this page will open. Right? Once that page opens, you can input your target sequence. Maybe at this point of time, you can put in any arbitrary sequence and please write down that sequence on your notebook once. Whatever sequence you're inputting here, please write that down on your notebook also. It needs to be uploaded in the FASTA format. Once you put the input sequence, the design tab on your right, it becomes clickable. And then you click on it and in the results section or the results will be shown below in a similar format.
So Suraj is asking that he does not have a check your own design feature, this thing. Um,
It is there in the middle. There are 3 tabs on that page. Don't go to design checker. Go to design custom guide RNA.
so Kirtana is saying that they are not getting those tabs.
You're not getting those tabs.
Maybe they are clicking, they're going to a wrong link. So I think I can unmute one of them.
Wait. What I'll do is,
else who is also facing the same issue?
So can you all see my screen now?
Yeah.
Yep. So I will try to, show you from here. So I will go to IDT-DNA homepage. Fine. Then I went to products and services. I scroll this down. CRISPR genome editing. Fine. Then I scroll down. Okay, it is slightly different. Okay. Oh, this page seems to have changed. Yeah. So actually there is one page before this that you need to click. So once you do products and services and then you go to CRISPR genome editing, then this page has been modified. So the first option here, that is Alt R CRISPR Cas9 system, please click there. Right? And then whatever page opens, you please scroll down. Then you will find this blue box, which is check your own design. Got it? I click here.
It's much better now.
Yeah. I click here and now I get these 3 tabs. Search for pre-designed gRNA, design custom gRNA, or CRISPR Cas9 gRNA checker. So I will click on design custom guide RNA.
So Suraj and Kirtana, I have unmuted you also. If you want to ask any question, you can ask directly. I hope that you are clear now. So they have Kirtana saying that they have got it now. So we can proceed.
Got it now?
Yeah.
Got it? So when you go to design custom gRNA, you go to species and you select your species of interest. It is by default Homo sapiens. Then input format, which is FASTA sequence, which is the default. And here I will put a sequence of my interest. So I have to name it followed by this kind of a, greater than sign. I will put any name of my interest and I will put an arbitrary sequence of my choice. Fine. Let's say I put hash greater than, say, genomics. And then I follow it up with some sequence in the next line. I am writing something in faster, my arbitrary sequence. Fine. Okay. Then as I enter the sequence, this design tab, it becomes clickable. I click on this design tab.
So by the time it is running, there's a question, Tani, that what
Yes.
design tool shall we use if we require to design cRNA for proteins other than Cas9, example Cas13 or Cas12?
Okay. So this site which I'm showing you, it is only for Cas9. There will be more that I will show you in the next few slides, whereby these questions can be answered, right? So now, when I scroll down, I get this kind of an output. I hope all of you can also see this for your own sequences.
Yeah.
Now, when you have seen this, when you have got this kind of an output, please have a look at this grid here. So the grid here shows in red the regions whereby this is, kind of quantifying the efficiency or the estimate, predicting this, the efficiency of the designed guide RNA in the form of off-target and on-target potential. So on-target is on the x-axis and off-target activity is on the y-axis. So in the ideal case, you want your best performing guide RNA, which is designed, should be falling in the quadrant depicted in white here. Because in the red region, you will have low on-target activity and high off-target activity. So on the x-axis, whereas on the y-axis, your high off-target activity is at the bottom. So your most desirable quadrant will be this one, where the white region is depicted. Now there are numbers written in these boxes. So if I go to this number 1 written in the white quadrant, it shows that there is one guide RNA which is falling in this white region. Fine. If I click on that, that is showing me the list here. Wait. Now, when I do not click on this, nothing is clicked, then it shows me the entire list of all the guide RNAs that are targeting this region, that have been found in this region. Now here, this is also showing this bar scale. It is showing me the coordinates as well as the genomic orientation of these designed guide RNAs. If I mouse over this one, this is showing the start and the end position, the design ID, as well as if you look at the grid right now, there is one number, one box, which is highlighted in green. So basically, this is the identification or the, what I should say, the activity score On the grid, it is highlighted here. Now, if I go to any other sequence, others are highlighted. Now, when I say that there is 3 written in this box, that means there are 3 such guide RNAs in this whole library which has just been designed, which are falling in this category of the activity score. Fine. If I click here, I will get a list of all those 3. Let's see. And the same list of all these is given here with the position, the sequence, the PAM site, and on target score and off target score. So first, let me explain to you what is on target score and what is off target score. On target score is basically, it indicates the predicted editing performance of the guide RNA at the intended target site. The higher the value of on target score, the better it is. And similarly, what is off target score? Off target score shows a higher value indicates lower off target risk. Now, off target score is the inverse of on target score. That means the higher is the off target score, the better it is. That means better will be the performance and lower off target effects will be seen. Lower off target activity. If you want to see details of this off target, that also you can see here. It is listed all over here. All the off target effects can be seen here. Fine. Then the PAM sites are mentioned and the score of the off target activity. This is mentioned. A lower score indicates higher potential editing because your intended function is higher editing. So lower score in this case is good. Okay. And the whole list has been organized in the increasing order of the score. That means the sequences at the top are better to be chosen. But we are not going to select this. This is, I've hidden the off target effects. Going back, I will look at the sequence again. How I will choose the best combination is I want highest on target score and highest off target score. More important is on target score. So I will select a sequence which has highest on target and off target score. Fine. Now, looking at this page again, you can also see a a design guide RNA of different orientations here. Fine. So when you select a particular sequence from here, you have to order it as it is. You do not have to change anything in this sequence. You do not have to include your PAM site. You do not have to do a reverse complement. You do not have to order in the form of RNA. For ordering as an RNA, what you will do, you will usually replace T with U. But all that is not required. You should order as it is. Now it is your choice. You have designed it here in IDT. You can order it to IDT, or you can copy this sequence and order to some other site or some other service and tell them that you want these as guide RNAs. Fine. So this can be designed for you. Oh, this can be synthesized for you. Let me take you back to my slides where I have done a better analysis of your sequence. So is everybody able to see this kind of a grid score, etc.? And if there is anybody who wants to share their results, why don't you just tell us what is the score or what is the sequence that you've got? Anybody who wants to demonstrate.
So I'm also starting a poll that if you were asking that you were able to design your guide RNAs. So so let's see how many students have actually were able. So perfect. At least 92% Tani at the moment were able to design. They were
Okay.
So this is a good result. So far, so good.
Okay. So
Perfect. 93% now.
okay, great. Very nice. Thank you so much, guys. Please pay attention again here. So for a best synthesis, for a best design, you want higher on target score and higher of target score. Fine. So I have shown that in the green spark here that ideal situation is both of these scores should be high. Now, like I mentioned, I am again putting it across here for everyone's understanding that please go to this, please pay attention that common design or ordering mistakes which lead to failure of the experiment. Now, many times what will happen, you will think that I have designed the correct, this thing on IDT platform. How can my experiment go wrong? Although your design was correct, but you made some mistake during ordering and you did not order as per the suggestions, that can lead to wrong guide RNA being supplied to you and which will fail your experiment. So what are those mistakes? First is including PAM site. You do not have to include the PAM site. Do not include the PAM site. Then entering the reverse complement. Oftentimes we will think that, okay, this is the sequence of the DNA. Now, since we need it in the form of RNA, we need to do a reverse complement. No, you do not have to do a reverse complement. Sometimes people will just simply do a reverse of the target. Even that should not be done. You do not need to do that. Whatever orientation it is showing 5' to 3', please go ahead as it is. You do not need to change the orientation also. You might think that, okay, the orientation for with respect to the target will be different. No, do not do any changes like that. Third is entering as RNA. Like I just said, that oftentimes people will replace the T's with U's and order it in the form of an RNA. No, do not do that. That is not required. Fine. Whatever sequence that you just got, you order it as it is whenever you are using in the experiment. Fine. With this, we are done with this one tool. I hope everyone has followed IDT. IDT by far is the best option for this kind of designing, especially for Cas9. Now
There's
coming to the yeah.
So Suraj
Yep.
is asking that what determines the cost of the sequence? As in yours, it shows 115 and his is showing 95.
Yeah. So there are many parameters, Suraj. One is the length of the sequence. Second is the GC content or the nucleotide content can vary. Plus the, these are, prices. I mean, my slides were made a few, 2-3 weeks back and the company may have changed their price list. That may also be the reason. Plus also what it matters is the quantity which you are ordering. Although I think yours is also default and mine is also default here, but in case you're ordering of different quantity, the prices would be different. And as you can see, it is USD here. I hope you're also looking at USD and not any other currency. Because on my site, when I was streaming from my browser, it was SGD, not USD. I hope that clarifies this doubt.
Yeah, I hope that too. So
yeah,
he's saying understood.
Yeah.
Great.
Okay. Thank you, Suraj. Yeah, that's a good observation. Thank you so much. Now, a few minutes back, we had a question from a participant that how we can, design for different Cas proteins. So for this, I told you that there are different tools. So this is another one very interesting tool, which is called ChopChop. I will post the link to you all. So I hope you all can use this link. Yep. So if you're using this link, you can open this site called ChopChop. And thereby, you have the option now, you can choose a different Cas protein. You have these 4 boxes and you can also choose the function that you want to get out of this, whether you want to do a knockout or a knock-in or what. Fine. Now, in this page, what you have to do is, this is the home page, what it looks like. So basically, when you choose the target, you choose any sequence of interest or you choose any gene of, genomic coordinates or the gene name. Anything that you want, you can put here along with the species name. and the genome version that you want to target. Then using which CRISPR-Cas9 system, whether you're using Cas9 or some other protein that you can choose, and for which function. Once you have done that, once you put the sequence in the target, you hit on find target sites. Fine. I have selected Cas9, and from there, this is the page. So you can select different PAM sequences here. Then guide RNA length also that you can change, whether you want 20 nucleotides or anything other than that. Other than this, I have kept all the options as default options. Fine. Once you've done that, do find target sites. Paste or type your target sequence in FASTA format and click find target sites. And there you will get all the target sites and you will get the sequences. Fine. Now I will take you to another tool here, which is, I think I'll give you the link again, which is so the third link that I've shown you is ecrisp.org. This is another tool, and here you can get a whole library of the crisp this guide RNA is designed, and you can have several advanced options. I am not going to delve in the advanced options today, but I will just show you how this is working. This is also just an exercise to let you know that several such options are available. There is this one from Broad Institute, which is also an important one. I'll tell you why this is interesting. Now, this one from Broad Institute, which is called portals.broadinstitute.org, GPP web portal. Now, here you have the option that you see CRISPR knockout, KO is knockout, or you want to do CRISPR activation, that is CRISPR on, or you want to do CRISPR I. So you can choose the functionality that you want, and you can choose your different CRISPR enzymes here, right? So as per the enzyme, your PAM site will also vary. That also is the option here. And then the target genome, which genome are you targeting? So the default option is a human, so that I have left as is, and then you can put the sequence here. Again, you have many options, whether you put raw DNA sequence or you put the gene name or the gene symbol or transcript ID, any of that is possible. Once you do that, there is also this option of pick quota. That is how many of these guide RNAs you would like to design. Now, sometimes you want to design at least 8 or 10 and you want to select the best yourself. But when you put some number like 5, so what it will do, it will design a library and give you the output of the top 5, which are, good enough. So then it will also show there is an option that report unpicked sequences. That means, although it will show you the first 5 which are picked, the rest of these which it could design, even those will be shown to you in a separate list. Fine. Once you get all this, you have the whole list, and depending on your own choice, depending on your experimental requirements, you can choose which one to go for. Now, there are some sites like this one, Synthigo, where there are pre-designed libraries. That means there are already existing libraries to some of the known genes. There are some advantages and some disadvantages also. Disadvantages are when you cannot do some kind of custom gene synthesis, custom RNA, gRNA design, but you can get a lot of more information about the pre-designed ones. Let's see how. I will post the link for this one also. Now, in this, what you do, you just click on launch first. Once you go to launch, you put the genome name, you put the gene name, whatever is of interest, and you put your nuclease name. Depends for which organism. Actually, this is also a limitation of a sort that only few nucleases may be on their list because this is a pre-synthesized, pre-designed library that you're looking at. And when you click on search, it will do all these functions. It will be locating BRCA1 in Homo sapiens, choosing primary sequence, early coding regions, potential targets, and calculating the score. Now, this is how the output would look like. So this will show you all of these. We found 13 targets and recommend only these 4 top ranked guide RNAs for knocking out BRCA1 gene in Homo sapiens. It is showing you the genomic coordinates. It is showing you the orientation, as well as it is giving you a sorted score along with the sequence. Now here you see you have got U. Now this is basically when you want to order from their site only. Fine. Now, if you're ordering from their site, you just click on this one only and you select it here and you order it. But if you have to order it to any other site or any other vendor, then whatever is their recommendation, according to that, you use the sequence. Fine. Now, if I choose to continue with these 4 guides, I will be able to order all these 4 here. Now, this is for the pre-synthesized libraries. Then this one is one of the most interesting that I like. I will post the link and I will share what are the good things about this one.
Okay, so there is a question from Sanju again, that how do we receive the end product after ordering gRNA from these websites?
So Sanju, actually, this is dependent on your, wherever you're working, which research lab you're working, depends how they are having this kind of, the understanding between the vendor, how they are supplying it. All that is dependent on those. When you're clicking order directly, that is basically for American customers because they usually, these labs, these synthesizers are already US based and they have tie-ups with these labs. So when you click there, it will get all the shipping information, etc., on the next page, and there you can provide the information. So that depends on your institutional contract or something with these vendors. So these are basically what I'm telling you is for the designing purpose. So you design on these portals and you order from anybody in India or any provider who is giving these things to your lab already. So these are designing portals. Now coming back to this portal that I was showing you, which is guides.sanjanalab.org. So here you can do some really interesting stuff. So you can design your own custom CRISPR libraries. How it works is you select the target genome, then you enter the gene or drag and drop a list, which is in a text file, and then you select the library size. You select how many guides per gene you want. I've selected 8 here. You're free to select 5 or 10 or whatever. And I've kept all the other options as, default options. Here, the data, whatever you design, you can get it in the form of an email. So if you put your email address, it will send you the entire design library to your email. So that makes it one step easier for you for the, for future reference also. Once you've entered this, you click on step 2. Fine. Then you can export library from this here, which I'm showing in the, on the left, there is an option, the .csv file, or the same option can be clicked from the top right also, where the blue box is showing, blue tab is showing export library. Now let's understand what is depicted on this page. So basically it's saying that BRCA1 has 8 guides in the library, which is targeting 3 of the 26 exons. That means BRCA1 has 26 exons, and the portal could design 8 guides. Guides, that means guide RNAs, which are targeting majorly only 3 of these exons. Now, this is showing the expression of all these in different, tissues. Actually, I'll show you in the next slide. So, if you look at this expression, which is shown as a bar graph, this is color coded. You could scroll over to a particular exon and see what is the expression in different tissues. Now, for example, you wanted to target BRCA1 gene in the heart of an organism. You will know exactly where the expression in the heart, or I mean, in which exon's expression is maximum in the heart for BRCA1, and you will probably target your guide RNAs against that one, right? Or similarly, if you wanted to target a different tissue or a different this thing on a in, let's say you wanted to do a BRCA1 study across different tissues, so you will design different guide RNAs for BRCA1 for different tissues, and this will be possible through this kind of information. So I hope this answers the earlier question that somebody brought up, that how you can select tissue specific guide RNAs. So this is one portal for that. Right. Now, once you've got this information, you please look at the output result. Here, the definition of on-target efficiency is slightly different. Efficiency scores are computed using the techniques described in the particular paper. So what you want here is maximum on-target efficiency and minimum off-target efficiency. So you will select some of the top scoring ones. The PAM sequences are also given that you can see here, and the functional domains are also given. Fine. So I particularly like this portal a lot because you can export everything on email, because you can get tissue specific information also. But the limitation is that you will only have these maybe 4 or 5 different tissues, not like every other tissue, but at least this much information is also useful. Then this is about the the sequences alone. Now, if you were told by your supervisor that you really need to give me the genomic coordinates also of the target sequence, maybe this is the option that you can choose, which is benchlink.com. I'll send you the link. Yep. So here you can get not only the sequence, as well as you can also get the strand orientation, everything along with the genomic targets. Then here again, you have the on-target and off-target score. So please, guys, mind you that whenever you are using any portal for synthesis or design, you please go and check on their site particularly what is their definition of on-target score and off-target score. Depending on their particular portal, their definitions may vary, and thereby their gradation of the score may also vary. So your selection will be different for each one, right? So I think that brings us to the end of all of these tools. I would like to know from all of you if you are a little bit more oriented towards how to design these. And maybe Diksha, if you permit me, I mean, if you feel like you could run a poll that which is their most favorite portal
Great.
for guide RNA design. Yep. And if anybody faced any problems, we are open to questions now.
Okay, so should I give all the options in Dr. Tani? Which option should I give them?
I think you leave it blank and let them write the answer, or you want you want to do a poll. So maybe you could do IDT, then Sanjana Lab,
Chop.
then GPP.
Okay, okay.
And Chop Chop.
Anything else?
I give you 4 options, right?
Yeah. So you have given me IDT, Sanjana Lab, Chop Chop, and the fourth one was?
GPP.
Yeah, GPP, yes. Okay, so just give me a second.
And maybe you can also have this option of Synthigo.
Okay, great.
Yep. So we have all the 5 options.
In a moment, I'll just create that poll and we'll ask them. So in the meanwhile, if any of the participants have any query, you can. So which was the last one? You Synthego, right?
Yep.
Hmm. So I'm asking the question that which one did you follow the most?
Yeah.
Will that be a better question, Tani?
Yep, that's fine.
Let's have the poll. So
Do we
please,
have questions in the meanwhile?
yeah. So we can have the questions. So we have maximum votes for IDT and the minimum number are for GPP.
I know. Yep.
So at least 19 votes are for IDT, 65%.
Oh, great.
The rest of them have very limited number. So most of them 20. So the votes for IDT are increasing.
Yeah, yeah.
Great. So
Wonderful.
let's have some questions at the end. So actually, there are a lot of questions, Dr. Tani. So what can we do is it will take quite a long time. So how do you want me to do? So should we take them or we should send the answers to them if you have time?
So let's take them now.
Okay, great. So let's go from the starting ones which we missed in the beginning. So
So I
I
think it
think
starts from Sanju's question. What does off-target indels mean?
yes.
So Sanju, off-target indels means insertions or deletions, that long form for indels. So insertions and deletions which get incorporated or which happen at an undesired location, which was not really planned, which are happening at an off-target site, right? And then, yeah, that's the thing. Then Diksha, next question.
So the next question is, can we develop a